guess you're all basic with the topic here. So I'm going to be talking about Reseek and related issues with Muscle-3D and so on. And I'm going to be kind of throwing, okay, now the slides on to advance. Here we go. I'm going to be talking quite a bit about Foldseek, which is kind of the most obvious comparable competitive method, whatever you want to call it. And I'm very well aware that it's notoriously difficult to benchmark and properly understand and represent competing alternative algorithms. So I just want to emphasize that I really did my best to do all the analysis here, but I very much would welcome any feedback, comments, suggestions from Steinegger Lab or anyone else who would like to dive in and check or look at these results. So sort of biology 101 here, you guys know this stuff. Proteins are linear chains, otherwise known as sequences. You typically represent them as a sequence of amino acids, but you can also think of them as a sequence of structure states. So now you're looking a little bit beyond the residue and seeing what its context looks like. And the sort of canonical example here is a simple three-state secondary structure alphabet where helix and strand are the sort of the primary secondary structures that you come across. Everything else is lumped into the loop category. And you can deal with this in sequence alignment framework by now you have 2 letters for each position. And if you do a conventional alignment score, so it's the sum of substitution scores plus some gap penalties. And now the substitution score generalizes from something like BLOSSOM. And now we add in a term for, whether we have a substitution or an identical structure letter at that position. And hopefully that improves the accuracy and sensitivity of our alignments. The latest, greatest incarnation of this idea is Foldseek, who introduced this alphabet called 3DI. And the way this was made was clustering the secondary structure, or clustering, it's a little hard to explain briefly, but you're basically looking at the local neighborhood of the residue in terms of the torsion angles and of the nearest neighbor in 3D space, and you cluster all those angles into 20 states. And this kind of captures both secondary and tertiary structure at that position. And now the substitution score has an amino acid term, and then you add a 3DI term with some weight that says how important 3DI is relative to amino acids. But if you think about it, representing structure with only 20 states, you're losing a tremendous amount of information. So think about a 4-state secondary structure alphabet with helix, strand, turn, loop. Now you have that for a neighbor and the residue, so that only gives you a total of 16 states, and you don't have room to say very much more about what's going on in the structure. So the Foldseek paper sort of recognizes this issue, and they looked at the dependency on sensitivity with alphabet size, and they pretty much reached a point of diminishing returns at 20 letters, which is kind of convenient because there's already a lot of 20-letter code out there. So why does it sort of flatten out there? And I think there are 3 reasons for that. First, well, the framework itself is kind of limiting. You're only looking at the secondary structure of these 2 residues. And if you look at secondary structure, there's really 2 main states. There's strand and there's helix. And then, of course, a loop could be almost anything, but you quickly reach a point of diminishing returns because there can be almost any loop, but with a low frequency, and you're not really capturing much more information by expanding the alphabet. And the other problem that you run into is the more letters you have, you start to get a very low abundance tail, and you almost never come across pairs of letters from different members of different places in that tail, and your training data just doesn't have enough data to give you good substitution probabilities for those rare states. So the idea here that I'm going to talk about is, instead of just looking for more states in a single framework, we're going to introduce more alphabets, and that's going to basically blow the lid off this thing and allow us to have as many states as we want. So this is the basic idea behind Reseek. And so we're going to add together multiple structure alphabets. So how do we make an alphabet? So the starting point is, it's a feature vector, and it's really anything you want it to be. So we'll just think about it abstractly for a moment. We have one or more integer or real numbers, but we need to convert that to an alphabet. So that's some kind of clustering method. For example, k-means is a very simple way to do this. And we want to keep the alphabet reasonably small to make sure that we have good representation for all pairs of letters for training. And then we train this matrix on a set of alignments where we believe that it's pretty solid that most columns are really homologous. And so, for example, you could use TM align, and you want sort of the TM score to be in kind of a Goldilocks zone. You don't want to be too low so that the alignment's iffy, and you don't want to be so high that the alignment is trivial. So you kind of go for that sweet spot in the middle where alignments are difficult but not too difficult. And the feature, now you can get really creative. It could be any property of a residue which is invariant under translation and rotation of the atom coordinates, because of course that's arbitrary. The coordinate system you're using to describe the structure is arbitrary, so it should be an invariant. And in the current implementation, which I regard pretty much as a prototype, because we're only just starting to explore this idea, but there's something like 14 features, I think, of which 8 turned out to be more or less optimal. Those are the ones that are used by default. So there are 8 features, 8 structure features plus the amino acids and the 16 letters for each of the structure alphabets. So this gives us this large number of states, something around 10 to 11 possible states for a residue, and that's what I call the mega alphabet. And there's lots of scope for inventing new features here. That's sort of future work to explore other possibilities. For example, we're only considering the alpha carbons at this point, and we're only considering features which I could easily calculate. So for example, things which are trickier, like solvent accessibility and other things you can get out of DSSP, those are not included at the moment.
Is disorder propensity one of the features?
That could be, yes, absolutely. That could absolutely be a feature.
So I'm just thinking there's a huge amount of research that's being done on like substitution, or that has been done on substitution matrices for disordered proteins. Like their evolution is intrinsically quite different than the evolution of most regular proteins, because they don't have the same constraints. And I only say this because there's a very easy predictor that if the PLDDT is below 50 per residue in AlphaFold or CoLab fold, then you can essentially ascribe that as being a high confidence disordered region, right?
Yeah.
So if you're able to treat those separately, I think that right away would be a very big way to segregate the structures, and then thereby the sequence search.
Yeah, I think what I would do is I would bin the confidence score into 16 bins, and that would be an informative feature. It would also tell the algorithm, maybe there's some regions you can't trust. I mean, the algorithm would simply learn what is informative about PLDDT. So that would absolutely be a great feature to try. But the downside is it has to be a predicted structure. You can't do it with a PDB crystal. But yes, this is absolutely a feature that you could think about using. So this is just kind of to make it concrete. It's scorpion toxin, which is always a nice little structure to play with. And these are some example alphabets that comes out of Reseek. So now the substitution score is a weighted sum over the substitution matrices for the alphabets that you've chosen to use in a particular context. And now by doing this, we have a rich representation of structure as discrete states. And we can apply all of just any sequence alignment you can think of, any sequence algorithm you can think of pretty much, we can now apply to structure. So it could be Smith-Waterman, Needleman-Wunsch, blast, HMMs, muscle. It's really, conceptually, a very simple change. We just plug in the structure alphabets where we calculate the substitution score, and you're pretty much done. So what can we do with this idea? Well, it's one of the biggest advantages is that it's a lower complexity representation of the structure. So we've literally reduced it by one dimension. And from an algorithm development point of view, that's a huge advantage because you can do dynamic programming. You have these L squared, N squared type algorithms, and then you can optimize those. Whereas when you're trying to work with the three-dimensional coordinates, those algorithms are typically much more expensive. DALI is known to be NP complete. I would guess that optimizing the TM score is quite likely also NP complete. And also because we've sort of naturally integrated amino acid similarity together with structure features, it sort of increases the likelihood that you're going to get really homologous residues in a given column. So you're going to have fewer sort of obvious misalignments. So for example, DALI will sometimes misalign the catalytic residues in RDRP. And this is much less likely to happen when you're considering amino acid similarity as a feature. So now we need to do some validation and see how well this idea works. And there are really two sort of distinct applications of sequence alignment. One is to pick out segments, sort of local regions of proteins, and try and predict whether or not they're homologous. And the way you do that is typically to make a local alignment with a program like BLAST or Diamond or an HMM. And the accuracy of homology prediction, that's important for predicting the function of the sequence that you're looking at. At a more fine-grained level, you may also be interested in predicting which residues within that region are equivalent. So now we're viewing a column in the alignment as a prediction that the residues are homologous or possibly equivalent in some other sense. And now the algorithms are typically global multiple sequence alignment algorithms like CLUSTAL or Muscle or MAFT that assume that you've done the work of finding the homologous segments. Now you want to identify the homologous residues in those regions. And here the accuracy is important for things like tree building. That's probably the primary application. So how do we benchmark homolog detection? And here there's a very well-established and really quite convincing protocol for doing that. And it's based on this test called the SCOP40 test. And SCOP is a benchmark. It's a big database of domains. And here domains means an independently folding region of a protein, which is believed to be inherited down a tree. So it's sort of a homologous region. So we don't mean something small like a motif, which the word domain is sometimes used in different senses. But anyways, to make this test, you cluster domains at 40% amino acid identity. And then they're classified by, I'm not exactly sure how they do this, but let's just say it's an expert classification with essentially three sort of levels. Family level means that you have similar structure and function, and because, well, that's a somewhat arbitrary category, but useful. A superfamily is a set of domains which are believed to be similar because they're homologous. And then at the top level, there are similar structures which are believed to be due to convergence rather than homology. So how exactly do you know whether they're homologous or not? Well, you are kind of guessing, but this is the best we can do, and it's generally considered to be a high quality database. So here is a sketch of how the results are typically presented. So what we're doing is we're making a plot to show sensitivity against errors. So there's always a trade-off by setting a cutoff in e-value or score. So the more sensitive we want it to be, the more errors we get, and that's the picture we get here. The sensitivity is along the x-axis, so we want to go as far as we can along the x-axis, and we don't want to go very high along the y-axis because that means that the errors are increasing. And here I'm going to set how high up the y-axis should we go, and my answer to this is that 10 errors per query is about as high as you would go in practice, because that if you have a perfect e-value, that would correspond to an e-value of 10. So let's look at some actual results. So this is BLAST, DALI, Foldseek, TM-Align, and Reseek on SCOP40 with superfamily as the standard of correctness. And notice there's a log scale for the errors. And you'll see that below 10 errors per query, Reseek is clearly the more accurate because it's falling below all the other algorithms. Now, the other way you can validate is, and this is work in progress, improving the way the e-values are calculated. This is currently not on GitHub, so it's work in progress, but I believe that there are further improvements possible here. The other, excuse me. So the other standard you might use is fold instead of superfamily, and now you see you have like a qualitatively different picture because Reseek is doing well at low error rates, but then it does increasingly worse at high error rates. So what's going on here? Well, this is my interpretation of that plot. So if you remember, folds are believed to be due to convergence. So if you're a member of the same fold and the same superfamily, you're homologous. But if you're a member of the same fold but a different superfamily, then you're believed to be convergent. And I think that crossover point where you see the red dashed circle, that represents the transition from being in the same superfamily to being a different superfamily.
Sorry, I got a question here because this is actually something that we kind of talk about quite often is like, how do you resolve that a structure? Because like people often will say like, oh, these structures align and then they're homologs versus saying these structures align and they're similar structures, right? Not actually homologs. Do you actually see, okay, well, let me walk you through my line of reasoning. If the fold is simple, then I can buy that there's convergent evolution, right? Like if we're talking about like beta sheet, and a loop or something. But if it's a very complex fold, like a beta barrel or say even something like a palm print, right? Where it's like those three motifs coming together, even if there's no sequence homology at all, if the folds are the same, I would still say that those are homologs. It's just homologs that are not like well beyond the midnight zone of sequence alignment. So do you have any way of like, say, stratifying the kind of complexity of folds and actually seeing like, oh, for folds that are highly complex, meaning that they have many elements, that you're actually still able to identify homology versus simple folds where you can plausibly say that it's convergent?
So this is a great question, and I'm not at all sure. I'm not necessarily sure that anyone knows how to do this. I think it's off topic. So, um, you can maybe people can maybe disagree about how I'm interpreting this chart. I mean, it's extraordinarily difficult if you don't see any. Now, when you say sequence homology, you mean sequence similarity, right? If you don't see sequence similarity, then how do you decide? And I think it's just a guess. And uh, I don't know how they did that in SCOP, and this is definitely a weakness in this kind of validation, but it's the best we can do. I mean, there is no magic that will tell you whether folds are homologous or not. I mean, something like a beta barrel, I would think, because it's so repetitive, I would think it's more plausible than something like a palm domain, but who knows?
Well, think of it like a jelly roll, right? A jelly roll is probably a very good example. That's like quite a complex. So I will just give you, so one, I would say you should kind of be cautious of like saying, oh, these are just convergent and that there's no evidence that they're homologous. Because I would say if the fold is highly complex, that is in evidence of like homology, not just similarity. But
What I'm saying is that SCOP says it's convergent, so I don't have to have an opinion. I can just cite SCOP for this.
oh,
And
okay.
actually,
Then pass the
and
buck.
actually, And actually, I could actually, okay, then that's, so I could actually do that plot, which is I could separate out same fold, different superfamily from same fold, same superfamily, and I'll bet that explains this crossover. Then the discussion is, well, do Scott know whether it's convergent or not? But that I can punt on that, right?
Yeah.
Because
Okay.
I'm simply using their assessment. My own, I don't feel qualified to know. I take the point. I mean, I'm skeptical that you can know, but yeah, that's the state of the art here. So moving on. So the other important issue is sort of computational resources. Reseek is, I mean, it's a little faster than Foldseek, but not enough to matter. It uses something like a fifth of the memory. And Foldseek used a different metric, which I'm going to talk briefly about. In their paper, they used what they call the sensitivity to the first false positive, and Reseek gets essentially the same number there. So now let's talk a little bit about e-values and scores. So DALI and TM-Align report their own sort of idiosyncratic score, but they don't try and convert that to an e-value, while Reseek and Foldseek do report e-values. So just to remind you, what is an e-value? It's the predicted number of false positives you would get if you set the cutoff at that value. So to contrast that with the metric in the Foldseek paper, for each query separately, they look at the list sorted by the e-value, and they cut that list at the first false positive, and then they ask, what is the total number of true positives that you're left with over the whole benchmark? And the sort of the problem I have with this approach is to do that calculation, you need to know the optimal cutoff for each query. And of course, in practice, you don't know that. What you need is some kind of prediction of where you should set the cutoff. And that's exactly what e-values are for and why e-values were such an important innovation in BLAST. And of course, we can validate an e-value because it's a prediction of how many errors you should see. So here is a log-log plot for the Reseek e-value against the actual number of false positive errors that you measure on the benchmark. And this plot looks really suspicious, honestly. I mean, the fit is just ridiculously good. So is it really true? My best evaluation, I really worried about this a long time. So far, I can't find a problem with it. So it is tuned to the benchmark, but that's also true of Foldseek. I mean, there's really where else could you tune it? But there are only two parameters that I'm tuning. And I checked it by cross-validation. So I split SCOP 40 by superfamily. So a superfamily was put in one half or the other. And if you fit to one and then do this plot on the other, you get similar parameters and the same kind of good fit. So unless someone can find a problem with it, I believe this result.
Is there a similar test to the Scott 40 test on the CATH database?
Is there a similar test for what, sorry?
The CATH, so there's SCOP and CATH, right? CATH. There's like a
yeah, so you could obviously do all this with CATH, but it's built using DALI. So, so now you're biased towards DALI, you're hopelessly biased towards some methods and away from other methods.
It might give you some independent evaluation though, right? Because DALI, I tend to trust that DALI, like all things considered, DALI works remarkably well. And I think
let's see if you still think that in a couple of slides, all right?
Okay.
So if you do that same, so the orange dashed line here is what you would get with a perfect e-value, right? So this is the same plot, except now this is the Foldseek e-value. And we're seeing a very, very different picture here. So of course, it's very possible I made a mistake here, but I mean, this again, I was very skeptical of my own plot, and I really have tried to dig into this every way I could think of, and provisionally, I believe this result. And if it's correct, then the Foldseek e-values are underestimating the error rate by several orders of magnitude. So you can see I picked out one value here as an example. If we measure one error per query, the e-value cutoff to get that is about 3 times 10 to the minus 4. So now let's move on to multiple alignment. And if you remember, well, if you have a sequence method, all you have to do is change the substitution score. And yeah, you need to do a little bit with file formats and whatnot, but the change is really conceptually, it's a very straightforward and simple change, which Igor was kind enough to implement here. And so now how are we going to benchmark accuracy and scaling of multiple alignments? And with structure, it's getting difficult. So how do we benchmark sequence multiple alignments? Well, there's an objective standard, which is the residues should be homologous. Now, you don't know if residues are homologous, but you can use structure alignment as a proxy. If the same column is reduced in both sequence and structure, the residues are likely to be homologous. So you kind of benchmark sequence alignment using structure-informed references. But now we want to benchmark structure, so we have a more challenging problem. And I'm sure you're aware of the paradox of the ship of Theseus. So, you have your old ship and it keeps getting repaired, and sooner or later, well, you still have the same ship. And there is the less well-known protein of Theseus. And over long evolutionary time, things happen, it gets dinged around, it gets repaired. And at the end of the day, well, clearly the secondary structure is preserved, but the individual residues, is there any one-to-one homology with that very distant ancestor? We don't know. Maybe none of them are. Maybe it's only one or two. But just because you see all of that secondary structure conserved, it doesn't mean that you have a whole bunch of homologous residues remaining. So there's no consensus on how we should evaluate the correctness or the quality of an alignment. Residue homology is still reasonable, but it's not practical because we don't know it and we don't have an even better standard to fall back on. You can use things like RMSD, but that breaks down as well when you have more distant structures. So what to do. And the least bad option that I was able to come up with is to use this benchmark data set called Balibase. And this goes back to actually the late 90s, but the latest version was released in 2005. And it's long been the de facto standard for validating sequence alignment algorithms like muscle. And at the time it was built, the structures were known for about 18% of the sequences. Now we have predictive structures for most of the rest. And so we built this sort of enhanced database where, with the cases where we have known structures, and we threw out all the sort of easier ones. And this left us with 197 sets where we have full-length proteins with one homologous domain plus maybe some flanking domains. And we're just going to stick with the original amino acid reference alignments that were made back in 2005. The accuracy metric is the same as the original, which is the fraction of columns in the reference alignment that are reproduced by the tested algorithm. You can sort of summarize the accuracy as the average TC over the data sets. And then there's a statistical test to give you a p-value to ask if there's a significant difference between algorithms, which they don't necessarily have just because the TC is higher or lower. And here are the results. We're distinguishing methods as being structure or amino acid sequence based. And you can see Muscle-3D comes out at the top. There are two variants here, one with parameters that just were inherited from Reseek on SCOP40, and then with parameters retuned to Balibase. Then there's a couple, two or three sequence methods which are chosen because they're accurate or scalable or both. That's clustable omega, this thing called UPP2 out of Tandy Warno's lab. MAFT, of course, is well known. And then US-align is this kind of new multiple structure alignment method, which I think came out of the same lab as TM-align and is kind of based on the TM score, which was kind of claimed to be the most accurate structure method in Nature Methods 2022. And the ranking that you see here from top to bottom is statistically significant, except for the last two. That's US-align and MAFT, which are tied. So you see, just because you're using structure doesn't sort of automatically give you an advantage, at least if you believe Balibase is a good standard. But muscle is giving a large advantage. So I think there must be something real going on here, but it's very debatable what Balibase is really measuring. Unfortunately, again, it's kind of like with SCOP, there are some obvious questions as to what we're really, how good is the benchmark, but it seems to be the best I know how to do so far anyway. So also we can ask, well, how do these methods scale? And so notice the log scale for the time, and we also have a log scale for the number of structures. So this is basically a log-log plot, which looks nice and linear. And you'll see that Muscle-3D is right at the bottom of this plot. It scales really exceptionally well. Everything else is slower. And here is an anecdotal example that we're all probably very familiar with, and this is that alignment of all virus species. And here we have sort of a special case where we can assess misalignment even over very, very large distances because we can find the catalytic residues. And when we do that, if we look at the classic Wolf 2018 MSA, we find that more than a quarter of the sequences have misaligned catalytic residues according to the PSSMs. If we build the alignment using Muscle-3D, this is reduced to 3%. And now we're sort of down in the noise where, well, number one, I didn't even try to eliminate the permuted domains. So I forget how many, but maybe a third of those misaligned sequences are simply due to having a permuted domain. So there's no way to get them all right. And some of the rest are the residues where the PSSMs make funny mistakes. I mean, there's also some residue there in the errors on Wolf, but if you do a manual analysis, you can see that in the case of the Wolf alignment, the vast majority are correct. And with the muscle alignment now, we're getting down to where the alignment, it's kind of hard to find any remaining errors. And again, it's very scalable. This alignment didn't take much more than an hour on a 32 core machine. And as far as I know, it's the only structure alignment method which can align thousands of structures. And I believe this should scale to at least tens of thousands, but exactly where that limit is something that we're continuing to work on. So let me talk a little bit about this thing called the micro alphabet. Now, we just talked about a very large alphabet. It's kind of handy to have small alphabets for various reasons. You can do K-matrix, you can do SAMD parallel alignment tricks. And for several of these things, I tried to optimize the small alphabet, and they actually all converged on the same alphabet, which I call mu. What is mu? It's actually made of 3 smaller component alphabets, which are basically the secondary structure of the residue, the secondary structure of the nearest neighbor. So this is kind of like in Foldseek, except we've explicitly factored out those 2 terms. And then there's the distance to the reverse Euclidean neighbor. So it's saying, okay, where's the Foldseek nearest neighbor? Go down the chain in the opposite direction. What's the nearest neighbor now? And so for whatever reason, this alphabet keeps popping out. So there's something special about it. And from now on, I'm going to be strictly talking about vaporware. So these are ideas, half-implemented stuff, vision for the future. And one of the things we're working on, so this is with Evan, is trying to predict mu from sequence. And I'll talk about why we might do that in a second. But if you look at the way mu is constructed, there are some easy parts, and that's secondary structure. Local secondary structure is easy to predict. Then finding things like nearest neighbors or the distance to a nearest neighbor, that's really hard. That requires folding. So at least we've managed to factor the training problem into easy and hard parts. I think that's going to really make the training sort of easier and more successful, hopefully. And this is just kind of a historical note, but it's kind of cute that as far back as 1988, that's 36 years ago, there was kind of a neural network which was predicting secondary structure quite well. I'm sure with all Evan's tricks, we can do much better than that these days, but certainly a very small, we don't have transformers and 128 layers and everything here. We have a very, very small and simple, fast network which can give us the secondary structure. So it means that we'll be able to plug things like this into a purely sequence method, enhance it with predicted structure, and get improved sensitivity at very low additional cost. So that's kind of the vision here. So at a minimum, this sort of approach is going to work very well for local secondary structure. But if we're successful, we might be able to get a fast approximate large language model or something like that, which can predict all of mu and therefore incorporate some secondary structure. And this means that anything that currently looks like sequence-sequence alignment can be upgraded to sequence plus predicted structure, where we're using mu or something like that to represent structure. And this means that those minor changes to algorithms like BLAST, and muscle can integrate structure with a very small cost and speed, and we don't have to go and fold everything with AlphaFold 2. We can do this all on the fly with low overhead. That's the hope. Another direction is we can think about, well, now we have these discrete state models. All of the machinery that's currently applied to maximum likelihood for amino acid can potentially be applied to structure states. And we might be able to improve maximum likelihood tree estimation. I'm not sure if this is even theoretically defensible, but it's something to think about, and that's just kind of on the back burner to-do list right now because it's so extremely hard to show that your tree methods are really doing anything better than existing methods. So benchmarking is almost hopeless here. Another thing you can do is all the sort of technology in Hammer and PFAM generalizes very straightforwardly to this framework. So you make a seed alignment with Muscle-3D, and now we have this alignment conceptually with these 8, 9 alphabets. So every match state or insert state is now inserting a letter from each of these alphabets, and we can train those frequencies and posteriors, blah, 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 in all the same way. The only thing that's unclear without actually doing it is whether there's some very clever speed optimizations in Hammer 3, which are using SMD and whatnot. I think it's unlikely that you're going to be able to do all of that with the full mega alphabet. You might be able to do it with mu. So, you may be sort of going back to the old Hammer, which was much slower, but everything else certainly should be straightforward to implement. And then we should have a really substantial improvement in the sensitivity of HMMs. Another idea which we bounce back and forth from in Serratus and Palmfold and so on is what can you do with this thing I call it contact map profile. So you're familiar with contact maps if you have a single protein. But now if you have an alignment, a structure-informed alignment of a whole bunch of proteins, you can make a contact map for the sort of the conserved columns in that alignment. And then you can ask, well, how much do the distances vary for each pair? And the more that they vary, the less that they vary, the more that those distances are constrained. And so that implies some kind of functional constraint. So you can see the bottom left of this picture is the conventional contact map. So green is close, red is far. And the top right triangle is the standard deviation. So the light colored areas are constrained and the darker areas are less constrained. So hopefully by looking at a picture like this, you can pick out motifs within the domain that are doing something functional. And also over a larger scale, hopefully you can pick out domains, separate domains from each other, because if you have two domains that are doing different functions, they're not constrained to be at any particular distance relative to each other. They just have to fold together as a unit. So you should see like the whole intra-domain part of this plot being dark. Will that work? I have no idea. That's vaporware future work. And so if it does work, then you can do de novo domain identification. You can just take the whole of AFDB, cluster it, align it, cut the domains into pieces, and repeat. I think some charts that might work. And then you're going to get a 3D PFAM with those structure-based HMMs. At a minimum, you can do that for stop domains, so that's a given. And if any of this idea for finding de novo domains, you can throw those in the box. And this is going to give you a sort of a next generation PFAM-like library, which is more sensitive and covers a larger fraction of the protein universe. So that actually looks like we wrapped up pretty well after 50 minutes here. So I'd like to thank Igor for really diving in and doing a lot of hard work here on integrating Mega into muscle and doing benchmarking and parameter tuning. And that was it. Thank you very much.
All right. Any questions from any of the online participants?
Ryan has his hand up, I think.
I think he's clapping.
Oh, he's clapping. Okay.
Yes.
Yeah, I was clapping.
Okay. Otherwise, stunned silence.
No, I think that like you're kind of going to be gunning for, I think, what everyone is going for. These are all very low-hanging fruit from what we've seen with AlphaFold. So the question is like, what's the thing that can be accomplished the fastest? Like, what's the low-hanging fruit? And like, how do you get to that point as quickly as possible? Right? Because
Right.
I think anyone that's in this game kind of sees the same end goal here, right? Do you see what I'm saying? Like it's
Oh no, sure, and you can sort of see from the papers coming out of Martin's lab that they're sort of heading in similar directions, and I'm hoping that there's some synergy there and some things we could collaborate on rather than sort of quote unquote competing on. But yeah, that all remains to be seen.
but it's not just Martin's group, right? There's groups that are going into like AlphaFold DB and doing like protein domain segregation, protein domain prediction. A lot of ESM folds work was on trying to cluster proteins, right? Using the embedding space, which is kind of getting at the same issues here. Um, I
Yeah, I
mean,
think the decisive advantage that I have here at this point is there's nothing like Muscle-3D. So I can align 1,000 structures and nobody else can. That's kind of my take on it at this point.
so, but then, so like once you have multiple sequence alignment, like are you essentially, I mean, essentially once you have rapid search, you have the ability to cluster, right? Which is like
Yeah,
pairwise
but you can't. But okay, imagine you have full-length RDRP and you don't know about the palm domain. So you cluster them, but now you have one cluster, but you're not separating the palm domain out from the N and C terminal domains. So the thing that the structure alignment for a lot of proteins solves is how do you separate out the multi-domain protein?
I see. So you would be looking at like essentially like motif in the same way, like picking out motifs from the
Right. So
MSAs.
I'm scaling that up. Now you could maybe also do that with these sort of, what do you call it, like make a graph out of the local hits and so on. So it's not, but I think that having these very large structure alignments potentially has a real edge there. But like I said, this is all future work. We don't know.
How much like how much non like, but you're also making an assumption in pretty much all the work that the linear sequence is not changing, right? It's like the permuted domain. So any type of permutation, or if like there's structures that arise from loops or internal changes of the linear sequences, that all gets thrown out.
Well, okay, so
But DALI won't throw those out.
the most common way that happens is a circular permutation, not like in RDRP, but like a whole protein circular
Mhm.
permutation. And it's actually straightforward for muscle to do dynamic programming, allowing a rotation. So it's like circ uclust. So you can do the same trick, and that's also on the to-do list. I mean, there's a long, long list of things you can do here. My guess is those are rare, sufficiently rare that I should focus on other things, but I don't know.
Yeah, I don't know. Well, I think that that's kind of thing is that it's not unlikely that very few people know how rare or not rare it is because they're hard to detect.
So, well, yeah, okay, so that maybe that's a reason to do it then, because then we can go measure it.
Yeah. Okay. So what do you see as the main like what's the application? What do you want to go for with clustering or how are you going to tie this together to
That's my
like
question for you. That's your job, Adam. I build the tools. You do the grandiose, applications with them.
well I will have my I can have my fantasies about this, but I think we were talking about this with Evan, doing all viral proteins, like if we consider viral genomes and then as like a workbench, then being able to actually do domain segregation and say the equivalent of like multiple sequence alignment on structures for all viruses seems like a pretty good first pass. The pitch kind
Actually,
of
you
being,
could. So yeah, maybe you can extend it to so with the virus genomes, you don't know where the cleavage points are, right? So you have the same thing. So you just align the whole thing and you use this contact map profile to cut out the proteins. So you have at that level you cut protein domain motif. I mean, that might work.
well, domain segregation is a very hard problem, right? So like that's, if you have a solution to domain segregation, I don't know if the contact map is going to be de facto a solution because independently evolving domains can like kind of like merge. Think of the bracelet, right? So like you have a palm core that's like your right hand, and then the bracelet is on some double-stranded RNA viruses. It's like a ring that sits on the edge of that. But the
Sure.
contact map between the bracelet and the palm core is actually going to be very, very tight because like it's kind of evolved to like fit into that pocket. So you're going to see that domain is like gained or lost the bracelet, but the contact map won't segregate out the bracelet from the non-bracelet. What you're measuring with the contact map is like when you have like a linker region, like if you have two, say, fists and a linker region, and then the two fists are independently folding, then the contact map will segregate those easily. I agree there. But if they kind of co-fold, I don't know if that's going to give you.
Well, yeah, but the way you know bracelet is separate is because it's not found in most of the RDRPs.
Correct. But that's not represented in the contact map.
Sure it is, because if you align, sure it is, because now we align, if you align all RDRPs, bracelet falls out of the conserved core, right? So it's not even there. So then I've cut out, it's in that, so I've cut out the bracelet in round one. Then you cluster what's left, and the bracelet will fall into one cluster, then it will be its own domain.
Okay, but and okay, let's think about this the other way now. With say RDRP, what about going to like reverse transcriptase and then DNA polymerases, right? You should be able to recover all of those essentially immediately. But what's missing in like this kind of gets to your ship of Theseus point, right? A lot of the changes from one fold to another is not discrete, right? And what we're looking at is like a continuous transformation of protein folds over long evolutionary times. And it's not that, like, obviously there's a wrong answer is wrong, but it's not clear when a marginal answer is actually still correct. That's why I was saying I actually trust DALI quite a bit more than what is in the reference databases, because, you know, when you go into distant homology, and actually that also ties into this Reseek e-value, even when, sorry, Foldseek e-value, even when Foldseek was giving me like high e-values, when I would inspect the folds that were aligning, very often they would actually be a correct alignment, even with like an e-value of like 0.5. But it's a crapshoot, right? Like you don't know, but there's still a lot there. So
But
I don't know.
well, but here in this, okay, but DALI is performing worst here on Scott up to until you're really getting to convergent folds. So I think you have a hard time explaining why, well, why is this so badly biased against DALI?
So that's for this figure, like from a presentation, I need to see the kind of false positives of DALI or like the examples of where what like the marginal improvement is in Reseek over the other methods. Because yeah, it's really hard to kind of make a judgment with just the curves without actually seeing what these look like.
What you're looking for should be done with an ensemble perspective, like every structure can have several statements, like sub-stable statements, and gradually change the residues can change the distribution like portion with the statement. And sometimes the marginal change will happen along this way. And that's what we are looking for. It's more than one single structural representation for one method, but
Like ensemble prediction? Well, I think ensemble prediction, yeah, is a different topic, which is like quaternary structure
Yeah.
changes, which actually that brings up another really good point, is that like we're biasing towards one quaternary structure. And often it's like the quaternary structure that we have in PDB, like crystal structures for it, because that's what, like AlphaFold hallucinates ligands very often, right? Why does it do that? Because it's trained to give you a structure. Like if a class of proteins are co-crystallized with a ligand present,
Yeah.
the AlphaFold predictions like they are predicted as if the ligand is present, right? Even though there's no reason for that conformation to be the one that it chooses to report. And so when we're looking at either predictions or crystal structures, where we're looking at a snapshot of a highly dynamic entity,
Yeah.
and how we represent like the dynamics of that's probably the biggest missing. I don't know who's going to solve. That's almost
People world is trying to do this.
Yeah.
Some new papers in ICML and whatever, but I mean, we can wait for their progress, like the next level of revolution to unfold.
Yeah.
And then we can use of them to solve the question. It's very exciting.
Yeah, it's true. Have you thought about quaternary structures at all, Robert?
I'm sorry, say again?
Have you thought about quaternary structures? Like, do you see any way of incorporating quaternary structure into?
What's that?
Quaternary is the dynamics of the protein. So, you know, obviously primary, secondary, tertiary is the 3D snapshot that you get in one prediction.
So it's channel open, closed, all of that.
Yeah, exactly. Quaternary is the channel, like it's like, say, oscillating. In which snapshot state of the oscillation are you observing it? And can you represent like the ensemble of the entire oscillation? And really that's what you're looking for. Because you could make it probably, this is probably way outside the scope of what's going to be done in Reseek, but if you think deeply about like proteins, they're less stable structures. It's more about like, it's like a waveform of molecules that does a certain functionality, right? And so it's not about the one snapshot state that we observe. It's about the kind of landscape of possible states that this structure can take in like seconds of time, and that accomplishes something, right? And so if you're able to say which proteins have similar overall movement landscapes, that's probably the single most important metric, but no one's really going after that. And I think that's, it might be
the relevant paper I just mentioned in the channel.
Okay. But looking at maybe like something like confidence or flexibility may be able to play into that, right? Like if you know that there's more and less flexible regions, being able to accommodate that. That would be cool.
In this kind of framework, it has to be based on homology. So you could ask something like, is this channel homologous to that channel? Okay, yes, now we know the conformations it's going to take, but you're not going to do it de novo.
Yeah, it is. I mean, it's really based on the column alignment, right? Like you're using this very common column alignment approach. And I mean, it's the way these things are evaluated, but is that necessarily the best way to find structures that are either similar or homologous to one another? I don't know. I think like you state the problem very well. The ship of Theseus is the correct problem, and we don't know when something is homologous or not after, like, is it the same thing or not? And do you want to predict it if it's a different ship?
Right. So of course there's sort of a fudge here. So I'm having my cake and eating it. But I think that's defensible. So hang on, where's the protein of Theseus? Hang on. So it's this picture. So let's say, let's just imagine, and I think this is probably true for the jelly rolls, actually. I don't see any reason why any of the residues, like with palm domain, you have these catalytic sites, right? And so there's some very strong constraints in a very few places. But if you imagine the jelly roll, well, it has to make this complicated multimer. So it's very strongly constrained to keep the technology, to the, what do you call it, the topology or whatever you call it, of the capsid. But that's not really a well localized constraint. So I think any particular site in a jelly roll, you can do an indel or a substitution. So I think my guess would be that that jelly rolls are a very good example of this picture, and that over deep evolutionary time, there's just no homology left. It's just gone. But still, but then why would the Reseek approach work at all? And the answer is that because there was homology down the line at every sort of intermediate step, we have conserved enough of the amino acid similarity. And then there are these things I didn't have time to talk about, but it's how do you build structural features into an alphabet? And I think that's a really fun problem that we've only scratched the surface on. But if you sort of imagine yourself sitting on a residue and kind of looking at the residues around you, kind of like you're in a planetarium, right? How do you capture that picture in a couple of numbers? Well, even though residue homology has been lost, you still have a similar astronomy conserved at the end of this process. And that's kind of what you're leveraging when you do the whole Reseek alphabet trick.
So that's actually exactly kind of what I'm getting at, is like, these are almost two independent problems for me. One is to say, like, yeah, these two things are homologous, and then a lot of what you're setting up is optimized to find like homologous structures, right? And saying like, okay, these are of the same origin. And that's fair, but there's a practical problem of saying like, say I have like a local structure, like say an alpha helix, and you have this planetarium view of the things around you, right? And you have, all of these secondary structures, and you exist in that context. So in a way, that problem is kind of a separate problem from homology. You're just saying, show me where there's an alpha helix that exists in the same biochemical overall context as the one that I'm querying, right? And that may or may not be in homologous domains, but it's in a similar biochemical context. Because then if you extend that, then you enter this like ligand prediction problem, right? Where you say, if your starting context is now no longer a secondary structure, but a small molecule, you would want to search which planetarium constellations does this small molecule fit into, right?
Right.
And I think that's in a way kind of a more interesting problem.
Yeah, and I think you could, if you just sort of wipe the slate clean and stop thinking in terms of Scott 40 homology and start thinking in terms of other questions, there's so much scope for how you want to design this alphabet. I haven't even thought about side chains and a whole ton of other things you could throw in there, just because, there's only so many hours in the day to write code, right?
Yeah.
But I think if you have a particular problem that you're interested in, and then you get creative about how you design these alphabets, you might be able to do quite different things. I mean, for example, you might be able to find convergent folds. I mean, if that's, I don't think that's very interesting, because typically, what can you do with a convergent fold? Almost nothing usually. But if you're interested in that for some reason, you may be able to design alphabets which are good at that and not particularly good at homology. So you can specialize here and do new and different things, I'm sure.
Yeah. Well, I mean, like, if you're searching for a function, then convergent folds probably are very interesting, right?
Well, yeah, I mean, Well, exactly. So why are you interested in convergent folds? I guess the answer is that they're false positives if you are interested in functions. So it's good if you can positively identify them. But I don't think this framework is going to give you that. That's just, I mean, where is the information? I
Yeah.
guess really you'll have to look for sequence homology. And if you can't find it, well, that's sort of an indication it's convergent, but it's not convincing. But I don't know what else you could look at. I mean, your idea that if it's too complicated, then this is the whole creationism argument, right? I mean, you've always got to be skeptical of that.
Well, there are de novo folds, right? Like proteins do evolve de novo, but there's like that number where it's like there's only something like 6,000 predicted fold or 6,000 observed folds in nature, right? Um, if that's and that seems to be true. And like
That would be really fun if we can really do this, chop AFDB into domains. Maybe we can do Serratus, there were 6,000 folds known in and 1 million known in, we just have, I don't think we really know what the tail of that distribution looks like.
yeah, the rare folds. I think that's the problem is that it's like there will be it's like the zip F distribution. There's gonna be a very long tail, very rare folds. Did you have a question, Alex?
Oh no, so what you say almost exactly, I was doing that during my undergrad a while ago. It is very interesting to study like the inverse problem of this, which is you take a specific fold and then you find like all the collection of sequences on these many different alphabets that can generate this fold. And there's a lot of emergence, there's a lot of very interesting things. And about these rare folds, so one thing that we use as well, all these artificially created folds that do not exist in nature, but we know that they fold so in a specific way. Those are like really good candidates to consider because they don't have this kind of like homology, implicit homology acquired through evolution, because they're all artificial, but still they you can infer a lot of like structural properties from those because yeah, it's too much like pure
characterized information. As long as they're not biased. So like I think it was AlphaFold or ESM where they said they like made a new GFP, but it had like 70% identity to GFP. And you're like, is that really a new GFP?
There was a good paper from this guy Baker where they just created they just like models how like completely new artificial folds and they just like put them out there.
Yeah. Yeah, synthetic folds from Baker's group. Yeah.
The hallucination work from Baker's group, they are trying to generate a bunch of novel folding like nothing resembles something in the nature. And the synthetic they have to do X-ray crystal structuralize, and it's true. Like people can constantly generating more and more new food from like no MSAs.
Yeah, well, Philip Kim works on this quite a bit too over here, right? It's protein engineering. Yeah. It's a giant field.
I think something that be observed from nature doesn't mean they cannot exist. And sometimes we simply didn't recognize it from the matter of genomes.
Yeah. So you don't have currently a database of Reseek set up for the AFDB or anything, right? That's like on the to-do list too.
Right. Well, I was hoping to collaborate with Martin on that, and they would be setting up a web interface. So that's kind of, I'm kind of waiting to hear from them on that.
It shouldn't be too hard. How big is the database? Like how big?
AFDB, 200 million.
Like in gigs?
So I don't know. I could work that out. I mean, that's.
Do you know, Evan? How big is AlphaFold database in gigabytes?
there's multiple databases.
The 200 million folds.
I guess.
But it's going to compress the Reseek database will be a tiny fraction of that.
Okay. Well, I mean, if we're only talking like sub 5 terabytes, it might be not too hard to build a microservice around it on the cloud while we have cloud credits. That's not impossible to do. So we'd be able to.
The database is going to be pretty small. I mean, I just think my strong preference would be to do it in collaboration with Martin, because they already have all the infrastructure set up. It's just like one different page conceptually. And I can adapt Reseek to read the database. Well, no, actually I can't, because we do need a Reseek database to have all the letters. But I think we should rule out that option first before doing it with your lab. But if Martin doesn't want to do it, then to really make this valuable to the community, I think there needs to be a free web interface, or else.
Yeah, absolutely. Yeah, I think that's like a given. Okay, I mean, for applications though, I would say like pan-virum folding and clustering, like this is kind of what Evan was working on just before you pulled him on the Mu stuff, right? Is essentially to create a structure or embedding oriented, MSA or like graph of all viral genomes and connect it to one another. Ironically enough, the Sullivan lab had a pretty good rough sketch of what like a graph-based virome, they called it vContact, so vContact 2, but they mainly use that for DNA phages, and they like group things by open reading frames and used HMMs. But I actually think that I've been thinking about doing that, but at the level of folds, and I think that would be a much more powerful viral structure database, because that would essentially solve classification and identification on contigs, because we would then be able to identify every viral contig in all the Logan data and then map it onto like the known viral space. So
that's a massive folding job, isn't it? What did I miss?
Well, no, okay, but look, if you're talking about like, I guess I'm assuming we have like mu acceleration, right? But say if we don't have it, but if we don't have it, then we would have the like radius of say diamond. Like if you have every single protein mapped out and its relationship to
no, for sure. If we actually get a fast mu predictor, then potentially, we have to see how sensitive it is, but it potentially is going to be a lot more sensitive than diamond at the same price.
Yeah. Well, even if it's not like, even if mu doesn't pan out, then you would still be able to use the like the pangenome for the viruses, and then you would be able to identify all contigs in Logan, even if they don't have RDRP or a marker gene, right? You would say, okay, well, here's a contig. It aligns to a known viral protein. It's this part. It has this linkage to an RDRP. And therefore, the identification of like the number of like viruses would kind of explode, because most viruses are going to be short. I maybe have to draw this out to explain.
I'm not sure I'm following. If this is purely sequence based, well, the problem with that,
until we get mu operational, you would use the viral pangenome as the scaffold. But to make that viral pangenome, you would use the structure maps.
Okay, so you really need structure for this, because everything that's not RDRP just evolves too quickly. So you really need some kind of structure.
Yeah, I think that like structure is where you would get the most bang for your buck, because that's when you would actually be able to go past that sequence horizon. Yes, you're right.
Yeah, well, that's part of the vision here, is to do a sort of a structure enhanced diamond, and I'm just very curious to see how big of a boost in sensitivity you get from that.
Okay, so what do you need from us?
A fast mu predictor, that's Evan's job.
All right, Evan, you got work to do.
I say this one I'm doing, wait, fine.
Any other questions from anyone in the group? All right, Robert, thanks for the talk. That was really good. You've been busy as always, eh?
I have no desire to play golf. This is kind of what I do.
Yeah. Well, I mean, there's good hiking, but yeah, I get what you mean. Okay. Then thanks. I'll post the video for this in the channel. And then I think just let me know when you're ready to do any kind of bigger computes and we can get this going.
Well, bigger computes for what?
Well, if you like to start actually, like, I guess the first step would be calculating all the structures for all the viruses, at least like the reference viruses. And then once we get that, then we would want to make multiple sequence alignments for everything to kind of organize, like, where are the domains? What do we see? And then we can manually inspect those. And then
I think that falls in your court. I mean, I have
I'm looking at
I have
Evan.
the tools except for the AI piece, which is Evan's working on. So it's kind of like you let me know what tools you need, and I've got them.
All right, sounds good. Will do. All right, take it easy, Robert.
All right, thank you.